guys, welcome back. Today we're going to go through dihybrid inheritance and Punnett squares. So here, pause the video, try it out yourself, and then continue on to see the explanation to the answers. Okay, so as I said, this is dihybrid inheritance, but how does one know that? It's because it involves two gene loci, aka two alleles, for the same characteristic, as you can see over here in those sentences. Now, something that you really need to pay attention to is whether the question here says if it's homozygous or heterozygous, because then that will tell you what the parental genotypes will be. And the reason why they're a mixture, as I've written there, is because it says it's heterozygous for both traits, tail and skin. So it'll be a mixture. Okay, cool. Now for the next one, the gametes. Gametes, basically, it's very easy to do. There's this whole method of in, out, of how you compare them. So, for example, if I went from big T to here, that's one pairing, or one gamete. If I go here, that's another pairing. And you just continue to do that until you get the gametes, really. So, next thing. It says the F1 offspring genotype. Now you might be thinking, well, what is this space for? You know, I know that you filled it, you've done a table, a grid, a Punnett square really, and you've gotten to the answers, but how do I know that I had to do a grid? How do I know I had to do a Punnett square? Well, it's because it's inheritance. You can't find out the offspring genotypes without having done a Punnett square in the first place. I mean, you can, but it's extremely difficult and defeats the purpose. So anyways, so whenever they give you space, as such here, or this whole space really, it's indicating that you need to, draw a grid. So, when it says F1 offspring genotypes, the way you can work that out is by writing the genotypes for both the parents, really, the mother and the father, on the top row and down as well. And in that way, you can just do a Punnett square, TD and TD will make TTDD, all capitals. It's very simple. If you don't know how to do a Punnett square, there are so many videos online of how to do it. So that's that. Okay. Then it says offspring phenotype. So genotype is the actual, letters, but the phenotype is the characteristic which is displayed and the characteristic we see, right? So curly or straight, stuff like that. So, I just matched it up, but you might be thinking, well, how do I know if it's curly or pink, Or curly and pink, really. Is where you look here. Big T is dominant to the allele for straight tail little t, meaning if there is TT, okay, let's just do this, that means that it will be curly because T is dominant over little t. But if it was TT, little t's, right, then this would be straight. Because big T is dominant over little t, it means that little t, aka straight tail, can only be expressed if there's two of the lower cases. Same thing goes for pink skin and black skin as well. Cool. So we've got that sorted. Oh, one thing I forgot to mention is that when you do a Punnett square, or even when you write the gametes, I would strongly recommend you circling it, just like I've done there. I've always been taught it, and it's always written in the textbook as well, so I'd rather you do that than not do it and potentially lose a mark. I'm not really sure how the marks can really, what their opinion on this circling matter is, but I was always told that we should, so that's a little pointer that you should know. Okay, phenotype ratio, what is that? Well, when in class, the teacher's like, oh guys, you need to memorize the bunch of ratios, well, this is the time to shine. Offload that memorization into the page. So the reason why you can easily know what the phenotype ratio is by understanding really that it's dihybrid inheritance, aka 9:3 to 3:1. That's something you memorize, right? But if you didn't know that, then you can easily, it's not really easy, it's very time consuming, but you can work out the ratio from the Punnett square, but you do not want to be in that sort of position. Just please memorize the ratios. It will make your life so much more easier. Yeah, okay. Next thing, it says about how, this phenotypic ratio can change, or might be different really, if it's autosomally linked. So here the answer is like, the alleles have not completely remixed because of, like crossing over, right? What if, crossing over didn't really occur to its full extent? So in that way, the ratio would be different. And yeah, that is the end of today's episode. So I hope you've learnt a lot about Punnett squares and dihybrid inheritance. And try some more questions yourself as well. I will have a playlist on patterns of inheritance anyways, so you can go on that. Yeah, remember, there are 2 most important exam technique tips. 1, read the question first, and number 2, state all the obvious points. Now over to you. Try some questions and let me know how it goes.